very faint.
Yeah, there it goes.
Yeah. All right. Can anyone tell if the mic picks me up through the mask? Okay. All right. These need to go.
We'll ask some questions in small groups.
Cool. All right. I'm going to go ahead and get started. So similar to how I walked through the homeworks, we're going to walk through the pre-midterm exam questions, starting with this first one. Using this graph, so this is the reference graph of GC content and melting temperature, which of the following can be concluded? So first off, what can we conclude using a TM graph like this? What do we use it to deduce? Yeah. Which one specifically? Right, so this is related to GC percentage, right? So if we go to, in this case, all of the answers go to a TM of 90, and we're gonna walk through each answer individually because that's kind of the way you're gonna wanna approach these questions on the exam, is make sure each answer you know whether letter A is right or wrong, right? Because almost all of them are going to be multiple answer. So, at a TM of 90, the percent A in the sample is 25%. How do we derive that from the information that we're given? Yeah.
So if you look at 90 and what's given P, it's about 50%. I said about 45, but and then if C and G are like pairs, then you take that's 50%, and then A and T are not another 50%.
No.
You get 25 for A.
Yes. So that is how you would walk through that. Let me try to find a marker that works. Please. A little bit. Yeah, so you have your reference graph, right? You go over to the 90C, you sort of go up, you look across at the reference graph, and you see on this axis that we're at 50% GC, right? And that means that you have 25% G and 25% C. It also means that you have 50% AT, and again, because they are equal, you have 25% A and 25% T. So then we can walk through these, right? The first one, yes, the percentage of A is 25%. The percentage of G is also 25%. And then looking at C and D, again, at a TM of 90, the percent A is 20%, that's too low, and the percent G is 50%, that's too high. So you know that C and D are not correct, so you would circle A and B for this question. Yeah.
Is there always going to be an equal amount of A and T and C?
Yes, because they base pair to each other, you always have equal amounts of A and T and equal amounts of G to C. Yeah. Correct, right? So this tells you the GC percentage. If the GC percentage was 90, right, then the percentage of G would be 45, the percentage of C would be 45, and the percentage of A would be 5, and T would be 5. Yeah, so if your GC percent is 90, right, if 90% of your DNA is GC, what's the other 10%? AT, right? So this graph just tells you the GC percent. You subtract that from 100, and that's your amount of A and T. But A is equal to T, and G is equal to C. AT is not equal to GC. Exactly. Yeah.
So I actually thought it was 45 to look at the percentages added up to 100 how it is. Is she gonna tell us the percentages, or will there be little bits of offs?
No, you'll get something like this, right? If you look at it, right, if you follow 90 up to the location that it's at, it's about halfway between 40 and 60, right? This axis doesn't go up by tens, right? So halfway between 40 and 60 is 50, right? So it's much closer to 50 than it is to 40, which is what answer C would, or sorry, answer C would have to be if it was at 60.
No, I 45 and on the y-axis.
Uh-huh. No, it's going to be like this. Like it's all, you are not going to be able to get it wrong by mislabeling, right? Like clearly that is above 40 and below 60. The only answers that fit with above 40 and below 60 are A and B. Okay. Cool. Okay. So you introduce into HEK cells a human artificial chromosome, a HAC, that contained a sign RNA transposon sequence. Which of the following events could result in the sign mobilizing into a HEK cell chromosome? So what's the difference between signs and lines? Yeah. Yeah, so signs are shorter. They've lost something evolutionarily as they've been incorporated as transposons. They need elements from lines in order to carry out their functions. Given that, what do we think about A? Does A seem correct? An active line RNA transposon provides the proteins necessary for the sign to mobilize? Yes, right? So that's kind of the definition of lines and signs, okay? And then following that, how does a sign actually move around? Looking at B, does the sign need to be transcribed? I'm seeing nods. Why do we know that the sign needs to be transcribed? Yeah. Yes, so it is an RNA transposon, so there needs to be an RNA intermediate that is transcription, right? So A and B are correct, right? And then the sign is cut out of the HAC and inserted into a HEK cell chromosome. Is this true for RNA transposons? No, right? Cut and paste is DNA transposons, right? So you're not going to have that mechanism in order to transmit this sign. Oh, sorry. And then the region of the HAC containing the sign is bound by heterochromatin protein 1. Do you think this is consistent with what we've said so far? I see no's. Why is heterochromatin protein 1 inconsistent with what we're looking at here? Yeah. Exactly. So if it was associated with heterochromatin, it would not be transcribable, right? And we've just determined from going through the answers that this sign needs to be transcribed. Right? So RNA elements need to be transcribed. If this HAC that contains the sign element is heavily associated with heterochromatin protein, it will be in heterochromatin, it will not be transcribable. Any questions on 2? Okay, now we'll get into an assay, right? So we're looking at a DNase protection assay. Which of the following statements are consistent with the results of this assay to analyze chromatin structure, right? So what are we looking at here? What does the output of the DNase protection assay tell us? Yeah. Right, and which banding type tells us if it was open or closed, and why? Yeah, so a thin or blank band indicates that it was in open chromatin, it was not protected from DNase, and you no longer detect it on your final blot. Okay. So, moving through this, right? Part 1, is the Harringtonian gene in euchromatin in brain tissue? What does euchromatin mean? Open chromatin, right? So is it in open chromatin in the brain? Yes, right? So you've lost the band. Yeah.
I might be mistaken, but I remember from the class, it was said that the control was lag. That means it was degraded, so it cannot tell if it's hetero or heterochromatin. That's why I did not choose.
So that was a particular type of control, and it was the minus DNase control. If the minus DNase control doesn't have a band, then your assay isn't working, right? But here, all of the minus DNase controls do have a band. So if you don't put DNase, nothing is getting digested. You see a band every time. You are able to detect the region of DNA. You just aren't able to detect it if the DNA is destroyed. So it's in euchromatin in brain tissue. Okay, is it in heterochromatin in liver tissue? I see some no's.
Yes.
I see some yeses, right? It's a little bit of a confusing question because it depends on if you're using the drug or no drug. If you're not using the drug, the Harringtonian gene is in heterochromatin, right? Using the drug is an external perturbation. Ordinarily, this gene is in heterochromatin in liver tissue. If you just look at the left, that's the ordinary state of the gene. Okay, so is it in euchromatin in liver and heart tissue? Since we just ruled it out of being in euchromatin in the liver, you can immediately discard this, but it's also clearly not in euchromatin in the heart tissue because it's protected both with and without DNase. You still get it on the northern blot. Gene is in euchromatin. This is not correct. Are you sure it was this question? Because there's two questions that use this image. Okay, so the chromatin associated with the Harringtonian gene is more open in skin than in brain tissue. Is that true? What would more open mean? No band, right? If it's more susceptible to DNase, you get less of a signal on this northern blot. So it has a minor signal in skin and no signal in brain. So that one is also false. So the final answer for this one is just A and B. Any questions on that one? Yeah. Yes, exactly. So that's the next question. But you can still say that the gene is in euchromatin in brain tissue, right? Untreated brain tissue, that gene is in euchromatin. In fact, in treated brain tissue, that's also in euchromatin, right? So A is doubly true on both sides of the, yeah. Yes. If it's blank, it means it was in open chromatin, and it was destroyed at the first step of this experiment when you did the DNase treatment. So if in the drug it wasn't blank, that would indicate that the drug is doing something to convert that euchromatin into heterochromatin. Yeah. Yeah. So it's a little bit of a phraseology problem here, right? In liver tissue, which implies untreated liver tissue. So in liver tissue where you haven't given it a drug, in regular liver tissue, is it in heterochromatin? The answer is yes, because when you add DNase without a drug, you don't get any degradation of that gene. So it's protected by heterochromatin in ordinary liver tissue. Oh, it does say. It's underlined and everything. In the control, no drug conditions. Right? So you're only looking at the left side of this. Thank you. Okay. Which of the following statements are consistent with the results of the same assay, right? So now we're looking at the drug conditions. The drug has no effect on chromatin structure of the Harringtonian gene in brain cells because it cannot get into this cell type. Is that consistent with these results? Is there an effect on brain chromatin structure? Then it's consistent with these results. You don't have anything to show the explanation is correct, but this explanation is consistent with the results. I know that's frustrating, right? Consistency versus does this evidence support? As far as I remember, there's nothing that's that nuanced directly on the next exam. But if it says it's consistent, right? It's just, is the data possible with this explanation, right? If it says, does the data support, then the data needs to show something about the reasoning behind that. It's a little bit of a semantics argument. Okay, so if the drug entered the liver cell and caused a change in the, does the drug enter the liver cells and cause a change in the chromatin structure of the gene in liver cells? So yes, so we add drug and suddenly liver goes from heterochromatin to euchromatin, right? We've opened up that chromatin, we've made that, that gene unprotected, right, in euchromatin in the liver cells. The drug induced the formation of repressive chromatin on the Harringtonian gene in skin cells. Did we go from euchromatin to heterochromatin in skin cells? Yes, exactly, right? So we went from open chromatin that was getting degraded to completely closed chromatin in the skin cells. So this is again true. The drug is doing all of these things. And finally, the Harringtonian gene is more likely to be expressed in liver cells upon addition of the drug. How can we infer expression from this data? Similar to what we talked about previously, Antonio. Right, so degradation of the DNA means that it's more open. If it's more open, it's available for transcription factors. It's more likely to be transcribed. Okay, any questions on this one? You first. Yes. Sure, this is all of them. Yeah, so for A, there is no effect on the heterochromatin or there's no effect on the chromatin structure of the Harringtonian gene in the brain when you add the drug. The inference here is that it's because the drug cannot get into the cell type. You don't actually have data on this from this question, right? But it is consistent. That hypothesis is consistent with the data. You've shown from the data that the chromatin structure is not being modified by the addition of the drug. And then here it shows, here it says because it cannot get into the cell type, right? Again, you don't have data specifically saying that, but it is consistent with the data. The hypothesis is still consistent with the data. Okay. The Huntington's gene has these CAG repeats. If you get more than 35 of them, the gene product becomes aggregable, right? It can start making these aggregates. So you test the effect of a natural product on aggregate accumulation in neuronal cells that have the gene with 5 or 150 CAG repeats. Which of the following statements is consistent with the microscopy below? So, starting with A. How could we maybe infer from this whether NP represses transcription of Harringtonian? Yeah, go ahead. Yeah, so in the 150 repeats, right, so we don't see any change in the 5 repeats, which is kind of expected because it can't aggregate. But in the 150 repeats, you see all these aggregates, you add the NP, and then boom, much less aggregates. That's totally consistent with this natural product repressing transcription of Huntington's. And then next, the NP causes increased translation of the Huntington's mRNA. Why is that not likely here? Yeah. Exactly, right? So if we were getting more of that protein product, we'd be getting more aggregates, not less. The NP increases the transcription of Harringtonin, specifically in the cells with 150 CAG repeats. I think because we looked at A and said that it's likely repressing transcription, we can rule out C as well. And then finally, with D, the NP causes the formation of Huntington's aggregates in cells with 5 CAG. Is that true? Do we start seeing aggregates with this product? No, right? So the only one here is A. A is the answer for this one by itself. Yeah, so if B were to be true, we would see much more aggregates in the plus natural product condition. Right? So that is, it's saying the NP causes increased translation of the mRNA. If the NP was increasing translation, there would be more of the protein, and you would get more of these aggregates. But instead, what we're seeing is we get less when we add the NP. So B is inconsistent with those results. For the 150 specifically, yeah. Yeah. Yes. Yeah. Because there's less protein in the results, you can infer that these results are consistent with any way of turning off protein expression, transcriptionally or translationally. You don't know which it is, but it is consistent with those. Okay. Below is a part of the Huntington gene that contains the CAG repeat. What are the first 4 nucleotides of primers that we're going to use to amplify this whole sequence? Think about the direction your primers need to go, right? They need to point towards the region you want to amplify. And by point towards, I mean the 3' end needs to go towards what you want to amplify. So B is 1, right? So B is going to bind on this part of the top strand. So what we're looking at here is just the top strand of double-stranded DNA. B is going to bind right there. G, C, G, G, right? 3' pointing towards the region you want to amplify, right? So you make a 4 base pair primer right there. It's going to point inward, right? We can't do PCR with just 1 primer. So what's our other primer? A, right? So at the very front, right? In line with the coding strand, again, when you peel these apart, that's going to lay down right there and use the other strand as a template to synthesize towards the middle. So A and B will amplify the sequence, C and D are incorrect. Yeah.
For A, it was confusing. Like,
For A?
yeah,
I mean, it's correct, right? It's just TGCA. You can just read off the first 4 nucleotides.
it would be like flipped, right? It would be 5' to 3' to this side, right?
Are you talking about like how it's similar to C?
Oh, okay.
You had a question? I just know that this is the coding strand for the Huntington gene because that's where the CAG repeats are. It doesn't really matter. You're just amplifying this region. Well, you want to amplify this region. So you just need primers that point internally. So that's the primer that will bind to the top strand and point towards the CAG. And then B is the primer that will bind, sorry, A is the primer that will bind to the bottom strand and point towards CAG. B is the primer that will bind to this top strand that's shown and point towards CAG. So you need both to amplify the region in between. Yeah. Yes. Yeah. So in order to actually make PCR work, you need your 2 primers. This marker is really bad, so I'll see if I can make this go. That would be great. All right. So if we have a strand that's given to us, right, 5' to 3', and we want to amplify this region, right, what we're going to do is we're going to first recognize that this is actually a double-stranded DNA, right? So this is what's actually there. We're just given the top strand for convenience, right? And then our primers are going to be designed to match these sequences right here. And the reason for that is when you peel this apart, right? When you peel this apart, this primer is going to bind right here and synthesize this way. And this primer is going to bind right here to the bottom strand and synthesize this way, right? So because they have a free 3' end, they'll synthesize towards the center of the template that you want. So angle your primers so that their 3' ends point towards the region that you want to amplify. And as you do successive cycles of this, you will amplify just that region. Yeah? So yes, the sequence of the primer has 5' to 3' here, and 5' to 3' here, but in the answer key it's written flipped because we always order primers 5' to 3'. Like if you Google IDT and ask them for a primer, you have to give them the sequence in the 5' to 3' orientation. Yeah. Yes, so because DNA polymerase is going to extend off of that 3' end, these are pointing towards the center of what you're amplifying because that's the way DNA polymerase works. It extends off the 3'. Yeah.
So the primers like they will start with elongation, right?
Yes.
So they meet, and then they have the double strand.
They don't need to meet, right? So they're elongating on separate strands. Right? When you peel it apart, you elongate here, you elongate here. Because you took a double strand, you peeled it apart, you elongate on other strands. Okay. Any more questions on PCR? Yeah. Yes. Yeah, the 3' ends should point towards each other because you're amplifying the region in between them. Yeah. Yeah. I don't remember the exam credit structure, do you? Okay. It's partial. So everyone that you get, everyone that you're missing, you lose a point. And everyone that you pick that wasn't correct, you also lose a point. So if you picked 2 that were correct and there were 3 that were correct, you should get 1 point out of 3 for that question, or 1/3 of the points of that question. It might be the way Canvas auto-grades. It might be that Canvas just auto-grades anything that is not 100% correct as a 0. Yeah. No, you can't get less than 0 on a question. I mean, it's a valid concern. Like, if one of them is correct and you pick all 4, like, yeah. Okay. Gonna go on to this, and I'm gonna need my glasses back for this one. Okay. So you're amplifying this gene sequence from the previous question. I'll save you the trouble. The regions flanking the actual repeat part are 20 base pairs each, right? So your baseline is going to be 40 base pairs. So on the exam, you would need to count that, but I'm just going to tell you that for convenience, right? So then if more than 35 repeats in this gene is what causes the phenotype, is what causes the disease phenotype, who out of these patients are you going to recommend for genetic counseling? So first off, what is the length of the sequence if it had 35 3 base pair repeats in the center of it? Yeah. 145. So that is 3 times 35, which is 105, plus the 40 that's flanking it. Yes. So anyone above 145 base pairs, you would recommend for genetic counseling because they are likely to develop the disease. So, patient 1, would you recommend for genetic counseling? Yes, right? They have 175 base pairs here. That implies that they have more than 35 repeats. Patient 2. No, right? 100. That is not the threshold we just determined from multiplying 3 by 35, right? So that is not 35 repeats. I think it's about 20 repeats. And then what about patient 3? Yes. So because Huntington's is a disease of aggregates, if you have just one copy of the gene, it will still make that bad protein, and that bad protein will still aggregate. So even though this person has 2 different copies of the gene, one that doesn't have very many repeats at all, but one that does, you would recommend them for genetic counseling because they will develop the disease. Yeah. So the 145 is because our primers are binding at the very end, and that sequence is 20 base pairs on this side and 20 base pairs on this side, that whole thing is going to be amplified plus however many repeats. So with 35 repeats, right, 35 times 3 is 105, plus 20 base pairs on the right, 20 base pairs on the left, that's 145 total. Because the repeat is CAG, 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 right? So the repeated region is 3 nucleotides. Yeah.
So if we're given a number, what is 35? So we just use 35? Because I was just looking at how far it went in gel, which, you know, obviously has to be like big for it to have that many repeats. So it just says the closer it is to like 175, right? But I didn't do the math with it. Does that make sense? Like,
Yeah, it makes sense that you wouldn't necessarily need to do the math, but you're given that there's a threshold, right? You're given that there's a threshold at 35 repeats, right? So you can do the exact math and determine that anyone over 140 base pairs is at risk, right? And this may be important for a gel that has someone, say, at exactly 142, right? Like 1 repeat too short to actually be at risk. Or somebody who's at 145 exactly, who's exactly at risk. Like, so you're going to need to know how much or what the size is of an exact positive result on this band for Huntington's disease.
if they give us like a number, it's not just like oh, it's just like, you know, it's like put that in.
Yes.
Would it be possible if it was like 4? We would just like multiply by 4 then?
Yes, if there was 4 repeats, how long would the sequence be?
And then
Yeah.
since it's 145, we want to lower it to 145 as a threshold, right? Since like
Yes. If anyone is above 145, they're going to develop the disease because they have these repeats, and when the protein is made, those repeated regions in amino acid form bind to each other and start aggregating.
So the question, will we ask one question that has like such amount of repeats, and they just multiply that by 3 and see?
Depending on what the repeated region is. Here it's told to you that it's CAG with the N, right? See how it's in parentheses with the little N next to it? That's that repeat, that CAG, repeated N number of times. So in this case, if that's repeated 35 times, 35 times 3 is, that's what that sequence in the middle will be, plus the 20 base pairs on either end that you're amplifying. So that would come out to 145. Yeah.
So patient 2 is incorrect because it's not repeated, so we're fine at that.
Correct. Yes. It's not enough repeats to be at risk for the disease.
And then would we ever be asked to go above the threshold?
We have gone above the threshold. So like 175 in both patient 1 and 3 are above the 145. Yeah. If you have more than that number of repeats, you are also in trouble. Okay, so just to clarify, the answers for this one were A and C. Okay. You isolated a potential telomerase enzyme that uses the RNA subunit with 5' ACCC, ACC, ACC. What do you need to test if this is a functional telomerase enzyme in vitro? Okay, go ahead.
DNA driver, like the sequence that's complement.
Yeah, so which one of these is complementary to that?
This is
A, right? GGT, GGT, G, right? That's going to bind to the 3' end of that telomerase repeat. if you're reading from the 3', CCA, CCA, C, right? That will bind right there, and that will extend and continue that repeat with the jumping mechanism that I've talked about before with telomerase. Okay, so we need a DNA primer for telomerase to actually extend that, because telomerase is a reverse transcriptase that's using this RNA template to make more DNA. And then we need what? To do it in a test tube. Yeah. The telomerase is the polymerase. It's a reverse transcriptase. Yeah. B, right? We need the nucleotides. And how do you know it's B and not C? Yes, so C is RNA nucleotides. B is deoxy, not tyrosine, deoxy TTP, deoxy GTP. And the reason you only need those two is because that's all that is going to be added to this DNA sequence, right? The only nucleotides in the RNA template are A and C, so the only nucleotides you need in the DNA that you're extending are T and G. Yeah. No, B is DNA, C is RNA. Telomerase is a reverse transcriptase that makes DNA. It uses an RNA template. Yeah. Yes, deoxythymine triphosphate, deoxyguanosine triphosphate. Those will be the nucleotides that get extended, right? So because it's the triphosphate, the polymerase activity of telomerase will break those phosphates, use that energy to make a phosphodiester bond and extend the DNA. Yeah. Yeah. Telomerase needs a DNA primer. It uses RNA as a template, but it is a DNA polymerase that works off of a DNA primer. Yeah.
So if we use RNA as a template, we're going to be using RNA nucleotides, DNA.
No. So this is a reverse transcriptase, right? So we're using RNA as a template, and we're using DNA as a primer, and we're using DNA nucleotides as what is extended off of the DNA. In this case, yes, because it's a reverse transcriptase. Okay, so this is A and B and not C and D. Okay. So if BRCA1 is needed for repair of double-stranded DNA breaks by homologous recombination, which of the following happens when you've lost BRCA1 in breast cancer? Would you get an increase in the number of translocations in breast cells? What is a translocation? Yeah, so how might I stitch two chromosomes together? In joining, right? So if I don't have the capability to do homologous recombination, what am I going to do when I get a double-stranded break? In joining, exactly. So you lose BRCA1, you're going to get an increase in the number of translocations, because every time there's a double-stranded break, there's a chance for in joining to mix up chromosomes and fuse them together. Okay, and then similarly, looking at B, non-homologous in-joining repairs double-stranded DNA breaks in these cells. Yes? Yes, right? So just like we just talked about for the translocations, if we lose a gene that promotes homologous recombination, we're going to have to fix double-stranded breaks using in-joining. And then C, GT mismatches accumulate in the genome. How are GT mismatches fixed? Base excision or mismatch repair, depending on whether the cell is dividing or not, right? So, does the loss of a homologous recombination factor matter here? No. So even without BRCA1, these will not accumulate in the genome. And then finally, thymine-thymine dimers accumulate in the DNA, yes or no? No, they aren't going to accumulate, right? Because this is resolved by nucleotide excision, right? We haven't done anything that's going to affect the nucleotide excision pathway. The only thing we've done is affect the homologous recombination pathway. Yeah. Yes. Strong UV damage, different types of chemicals that will get in there and actually like chelate your DNA together, make two thymines cross-link to each other. Okay, so this one is A and B. Yeah. Base excision cuts a single nucleotide out that it recognizes as a mismatch. Mismatch repair relies on the cell to be actively dividing, and it recognizes which strand is the daughter strand, so which strand has less age, because you can look at DNA age based on like methylation marks and all sorts of other things in the actual DNA. When cells are actively dividing, there is clearly to the cell an original template strand and a new strand, and it will recognize the new strand when it sees a mismatch and cleave away nucleotides on the new strand and resynthesize that. So base excision uses different mechanisms to just cut out one base pair and replace it with DNA pol beta. Mismatch repair eliminates a section of the chromosome and refills it just like regular replication. Yes. Yes. Okay. DNA fingerprinting was performed on DNA from a cigarette left at a crime scene. Which of the following conclusions are consistent with the results? So whose DNA is on this cigarette based on the banding patterns? Suspect 1, right? So if you look at suspect 1, all of those line up. What about suspect 2? Also, yes. Suspect 2, every single band aligns with something that is on the cigarette. Clearly, the cigarette has way more bands. It looks like there's probably a mixture. You've amplified more. You've amplified more different DNA. And then what about suspect 3? No, right? Suspect 3, none of their bands, or very few of their bands, align with what's on the cigarette. And if their DNA was on the cigarette, you would see every band present on them also on the cigarette. Mm-hmm. You might want to rerun that, right? Like that's one of those where like maybe I made a PCR error, right? Because how is this made? It's a gel where I've taken a bunch of STR loci and I've amplified them, right? So a bunch of the short sequence repeats, the simple sequence repeats, right? You amplify those and you run them on a gel. And if I was missing one band, right, what would be my expectation? That PCR failed for whatever reason, right? So I would want to rerun that to make sure I get the number of bands I'm expecting for the number of loci that I'm testing. If you get one that doesn't match, most likely that person did not leave DNA at the crime scene. But it probably means that they are close family members with whoever did. Would that be considered on what? Sorry.
On the exam, like let's say in suspect 2, if one of them didn't match and we pull that upside down, so we'll be more.
It'll be much more clear than that. It's not going to be tricky down to like squinting at a one particular band, whether it's misaligned or not. Okay, and then are suspect 1 and suspect 2 twins? How do we know that? Their banding pattern is different, right? Yeah. They could be fraternal twins. It's highly unlikely that they're fraternal twins. Because at each locus, they have a, what would that be? At each locus, they have, I want to say a 50% shot of sharing one with their sibling, right? Because if at that locus, sibling A got it from mom and sibling B would need to get it from dad to not look the same, right? So for these 4 loci, which is approximately what is tested here, they would have to be different at each one. Yeah.
If it was like E for some reason, like are they siblings 1 and 2, would that be like a viable answer?
No, I wouldn't say that they were siblings. It's highly unlikely. And it would be, you would need a lot more data to actually definitively say that, though. Yeah. Go ahead. Yeah.
What if like they had the cigarette and then they had like suspect like I don't know like 0 and then they said suspect 1 and 2 like related to suspect 0 based off of like the cigarette? Like, would that?
You wouldn't base it off the cigarette. You'd base it off of the banding pattern of suspect 1 and 2. Like if you want to do paternity or fraternity, you're going to look at banding patterns between individuals that you've collected DNA from. You don't want to try to deconvolute a crime scene sample that has multiple DNA patterns on it. It's going to be difficult.
My question is that, let's say that there were twins.
Uh-huh.
What result should I have to say that
The exact same banding pattern.
so is that like
Identical twins will have the exact same banding pattern for this assay.
what identical twins? So if you have common,
Not identical twins would be the same as brother-sister, like just direct siblings, right? They will, on average, have a 50% match. Okay. Debating. Okay, I just want to remind you guys how we're amplifying these, right? What are my primers for the cigarette? What are they amplifying? DNA. What DNA? DNA left on the cigarette. What regions of DNA? It's very quiet. I have no friends. It's the SSRs, right? So all the short sequence repeats or the simple sequence repeats, whatever you want to call them, right? The ones that are chosen for amplification on the cigarette, right? What am I amplifying in lane 1? Yeah. Sorry, suspect 1. The same SSRs, right? So like, if you're amplifying, locus 1, 2, 3, and 4 in the cigarette case, you better use the same primer mix for suspect 1 and for suspect 2. Why? What if I amplified different SSRs? Yeah, it just doesn't matter completely, right? So we need to look at the same loci in each of these cases. We're looking at what do these individuals have as a sequence length in these specific regions.
so like if you use my different SR, we must have a interaction.
Right. If you were using different SSRs, you can't do any solid comparison between the lanes. Okay. Based on the results from an experiment designed to test the effect of inhibitors on DNA replication, which factors' be inactivated by each of the inhibitors? So, with no inhibitors, you have a regular synthesis of DNA. With inhibitor A, the 5' ends of new DNA strands still contain RNA. Why would something still contain RNA in replication? Yeah. RNA primers that aren't being removed. So which of these answers aligns with RNA primers not being removed? A, right? So if inhibitor A stops RNase H, those RNA primers aren't being removed. What would happen if inhibitor A stopped primase activity? No RNA primers would be laid down, which means you would have no DNA synthesis, right? Because that's the whole way DNA synthesis starts during replication. And what if inhibitor A stopped ligase activity? No fully complete strands. You'd have a bunch of fragments at the end. You'd have all the Okazaki fragments. You'd even have long leading strand fragments because even the leading strand needs to meet up with other leading strands or other Okazaki fragments and get ligated together. Okay. And then what about inhibitor B? Inhibitor B, you just see no new DNA strands detected. Is B inhibitor B stops helicase activity consistent with that? Yes. So you need to peel the strands apart in order to do this synthesis. So the answer for this one is A and B.
Sorry.
Yeah.
If it stops the primase activity, what's going to happen?
You get nothing. You will get nothing.
But we will start to have like a rival, but
No, if there's no primase activity, you can't start DNA synthesis because you won't have any primers laid down.
so we will start, like, we will stop right after the helicase unwind.
Right, but you won't see any result of that. Yeah. You might have gotten that option. Both of them are consistent with the data. Yep. Yeah, so inhibitor B, you get no new DNA synthesis, right? And the question is, which factors could be inactivated by each of the inhibitors, right? If inhibitor B inactivated helicase, this is exactly what you would see. You would see no new DNA synthesis because you need to peel those strands apart in order to do any DNA synthesis. Okay. Okay, the below sequence is from the lac operon, and the A indicates the +1 site on the template strand, on the non-template strand, sorry. Based on this information, which of the following are correct? So, which direction does the plus go on the non-template strand? 5 to 3, right? So we're heading towards the 3'. So if A is your +1, where is your +10? Yeah, down on the 3' on the C, right? So we have the +10 position is at a C, and we can eliminate D, right? So we're at B, right? And then between A and C, how does the RNA transcribed from this start? Yeah.
We'll go downstream.
Mm-hmm.
Feels like this, we need to match the non-template, but this is the timing we need to use your also.
Mm-hmm.
Because we don't have like an RNA.
Yeah, so the non-template sequence is the same as the RNA sequence because the template is complementary, right? So the RNA transcribed from this sequence starts with 5' A, U, right? So it starts at the +1, and it's that nucleotide plus the next one plus the next one. Any questions on that? Yeah.
So I thought that the primer had the 3 inside of it, so why would it be you flip it and have it be UAA instead?
This is the non-template strand. So that's the +1 location on the non-template strand. That's going to peel apart. RNA pole is going to bind to the other strand, the template strand, and make RNA in the 3' direction, starting from the +1 location.
Okay, so it's the same thing, just using the that's how you got A.
It's the same thing as what? Sorry?
So you make like the complementary strand to this one?
No, you make the you make the complementary strand to the other strand, which is going to be this strand. Yeah. Yes. Yeah. We'll see how good this goes. Okay. So I'm going to cheat a little bit for convenience. Okay, so this is your +1 on the non-template strand, right? On the non-template strand, the plus goes in the 3' direction because the non-template strand is the coding strand. It has the same sequence as the RNA, right? So this is going to be your +5, right? So we're going to go in this direction, right? This is going to have the same sequence as the RNA with thymine swapped out for uracils. The way this works, right, is there's actually a second strand here. That looks like this, right? This is going to peel apart, but this is still the +1 location. RNA pole is going to bind right here and synthesize RNA this way, because this is the 3' end. So the new RNA that it lays down is going to be A at the 5' end, then A, U, U, G, etc., down to the 3' end. It's going downstream, which is the plus direction. Yeah.
So the non-template strand and what's the synthesized going to be the same, which is synthesized one has both complement the same strand.
Exactly. They're both complementary to the same strand, so they must be identical, except it's RNA, so it's going to have uracils. Okay, so just to recap, this one is A and B. Yeah. Yes. On the template strand, the plus direction goes 3' to 5'. Yes, the new DNA or the new RNA that it's laying down is being built from 5' to 3'. It's moving up the 5' direction on the template strand. On the template strand, the plus direction is 3' to 5'. On the non-template strand, the plus direction is 5' to 3'. But the way the polymerase actually works is by moving up the 5' direction on the template strand while laying down complementary RNA that's going 5' to 3'. Because that has to be the direction of what the complement is. Yeah. Yes, you include the +1. The +1 is the first nucleotide of the RNA. Yeah. If this said template instead of non-template, you would count the pluses in the 5' direction, make the complementary RNA going that direction. I'll give everyone a moment to digest that. Gets very turned around.
Doesn't help that it's, by its nature, can be very unique.
Yeah. Did we said 120 is what we're going for?
150 is when the stream stops.
Cool.
We had 2 hours.
Yeah. Sorry. All right. So, if we're going to look at whether other sugars can regulate the lac operon, first of all, let's clarify what conditions we're working under, right? So we're working under conditions of no lactose and no glucose. So what's the state of the lac operon by default with no lactose and no glucose? It's repressed, right? So we don't have lactose, so the repressor is still there. But we don't have glucose, so CAMP is high, so cap is there. So the cap is binding, and so pol with sigma 70 is ready to go, and it will happily transcribe that as soon as that repressor is off. So this experiment is set up specifically to look at whether other sugars can regulate that repressor. Yeah.
We wouldn't need an M cell for this to make that decision, so we should just assume that the, like, you know what I mean? You know how she says that we can't prove without finding specific proteins, but operon question should be secure in that.
Yeah, with the operon, it's well enough established that you can assume that what you're looking at is something that makes sense within the context of the operon that you've learned, right? So In this case, the repressor is binding, and you're looking at whether these sugars release the repressor enough for mRNA to happen. So we're looking at a northern blot, looking at mRNA levels when we treat with sugars A, B, and C, right? So which of the following conclusions is supported by this data? Starting with A, sugar C binds the lac repressor and prevents it from binding the operator. Does this data support that? Do we get mRNA with sugar C? Yes. So do you think sugar C is binding the lac repressor? Why not? How does lactose regulate the repressor? It binds to the repressor and it releases it from DNA. So the repressor searches for a particular sugar, in most cases lactose. When that binds, it is released from the DNA and you get transcription. I would not say that. It's exactly what you would think it would do, right? It binds to the repressor and it gets released, right? So when you express, or when you have lactose in the cells, the repressor is going to bind that lactose and get released off. Exactly. The repressor is always bound by default. So this data does support that adding sugar C binds the lac repressor and prevents it from binding the operator and results in transcription off of this repressor, or off of this operon. Transcription of lac Z is repressed in the presence of sugar A. Is that true? Yes, right? Again, this is a northern blot. You don't have any mRNA with sugar A, which says that transcription is still being repressed. Sugar A binds the lac repressor and prevents it from binding the operator. No, right? Because that would have the opposite effect. That would enable the lac operon to be active with sugar A. And then finally, D, the entire RNA sample was degraded in the sugar B lane. How do we know that's not true? Our control rRNA is still there, right? So it's not that the RNA was degraded, it's just that there's a minor amount of expression with sugar B. Yeah. Yeah, all you need to know for this class is when you have high glucose, there's low CAMP. When you have low glucose, there's high CAMP. CAMP binds the cap protein complex, which then binds the cap region, the cap site of the operon, and improves the ability of RNA pol and sigma 70 to bind. Yeah. Sigma 70 will bind to RNA polymerase without the cap. Sigma 70 will have extremely weak binding to the lac operon promoter without the cap. Yep. So if sugar A bound the lac repressor and prevent it from binding the operator would be turned on because it's the repressor, right? And the operator is not on because you don't have any lac Z mRNA. Yeah.
Can you explain the glucose-lactose relationship?
If they're both high, the cell just wants to use glucose for energy, right? So if they're both high, you don't get cap binding. You get very weak expression off of the lac promoter because you don't have improved sigma 70 binding. If glucose is high and lactose is low, the repressor is going to be bound. If glucose is low and lactose is high, then cap is going to bind because you have high CAMP. You're going to get extreme amounts of transcription off of this because lactose is binding the repressor and relieving the repressor. Okay. So you do a promoter mutagenesis assay with the Huntington's promoter put into neuron or skin cells. And then we're going to try to deconvolute what is going on with this, right? So walking through it, in number one, when you mutagenize the minus 300 to like the minus 225 region, suddenly your expression level flips, right? You get lower expression in neurons and higher expression in skin. Then you mutagenize the minus 250 to the minus 175, and you have normal expression again. So what does that tell you about where that regulatory element is that is making the expression flip? It's a repressor in one and a promoter in the other. What do you mean by in one and the other?
The expression of neurons without any injection is 10, and it's 5 when it's viewed from negative 300 to negative 225.
Mm-hmm.
So there is the promoter element that was lost in neuron cells, and then with skin cells, it's the opposite.
Okay. Yeah, so there's some element that is positively regulating transcription in the neurons that is lost there, and is negatively regulating transcription in the skin that is lost there, right? So there's some element there that different cell types have a different reaction to. How do different cell types have a different reaction to certain different DNA sequences in promoters? How is that maintained? How can I put the same DNA sequence in my skin and in my neurons and not express all the same genes? Yeah. Different transcription factors, right? So there's transcription factor binding sites all over this that are regulating it in different contexts. So stepping through the answers, the minus 350 to minus 200 region, so just the region that is deleted or that is mutagenized in one that doesn't overlap with two, right? So that region can positively or negatively regulate expression depending on the cell type. Yes, that's just what we walked through, right? So that's what's happening there. If you only included the minus 200 to the plus 1 of the promoter sequence, the expression level would be 10 in skin cells. What would happen if I deleted everything upstream of the minus 200? Which one of these would it match? If I deleted everything upstream of there, it's going to be most similar to 1 because the effect of 2 is nothing, right? Mutagenizing 2 did not change it from baseline. So if I do minus 200 to plus, if I do just the minus 200 to plus 1, if I've deleted everything that's upstream of the minus 200, I'm going to look just like condition number 1 here. I'm going to have 10 expression in the skin cells and 5 expression in the neuron cells. Yeah?
I think I missed it when I saw the choice, the option. I mean, doesn't mean like when you delete it, doesn't mean that it has this black side blocking it, or just like.
If you only included the minus 200 to the plus 1 of the promoter sequence, that means the endogenous, like the original promoter sequence. Yeah.
No mutation.
No mutations.
Oh.
Yeah. Yes, the black part is mutated in each of these. Yeah.
So if we're including from negative 200 to 1, why don't we include that part of 2? Because you see it go from 10 to 5, and it's saying that it can keep at 10 for skin cells. For row 2.
Like whatever is affecting the transcription of these is happening in the minus 300 to minus 250 region, right? If you just mutagenize minus 250 to minus 175, you don't get any change in expression. So if I'm only working with the minus 200 to the plus 1, I've lost the regulatory element that's in 1. Whatever is controlling neuron and skin to be 5 and 10 gets lost in the minus 200 to the plus 1.
I was also looking at like 4 and 5, like I guess that's like later in the thing, but could you also use that to kind of denote that, or no?
No. So you're mainly looking at what is lost here, right? And you see that when you lose something that is in the minus 300 to minus 250 range, you flip the expression levels. Okay. So, last one. All expression is lost in both cell types when the minus 150 to minus 50 region is deleted. Is this true? No, right? Because this is all mutated in number 4 entirely, and there's no effect. Instead, what's causing all expression to be lost has to be something that's mutated in number 3 that doesn't overlap with 2 or 4. So it has to be in that really narrow region between the minus 150 and the minus 175. So there's something in that specific overlap region that is entirely responsible for driving transcription. Yeah. Yes. It's A, B, and C, I think, unless I misread it. Yes. I can't really hear you, sorry. So yes, the skin expression got increased, so there is a negative regulator in minus 300 to minus 250 in skin cells. There's also a positive regulator in that same region in the neurons, right? Because expression went down in the neurons. And it may be the same regulator. It may just be it has different levels in skin and neurons. Any follow-up questions on these mutagenesis assays? Yeah. Yeah, so for B and C, right, I'm basing it off the fact that when you mutagenize the minus 300 to minus 250, you get a change in expression to 5 in neuron, 10 in skin, right? And then when you mutagenize the next region, the minus 250 to a little bit more than the minus 200, you get no change. You actually go back to the original, right? That tells you that the only thing that's affecting expression in the whole minus 200 to minus 300 region is that early element that flipped that. So if I only put in the promoter from the minus 200 to the plus 1, I would be missing all of what's deleted in section 1 and a little bit of what's deleted in 2 as well. But we've determined that 2 doesn't matter. 2 doesn't change expression from the baseline at all. So the only effect that's going to happen in the minus 200 to the plus 1 is we've lost that early regulatory element that, upon its loss, flips the expression in skin and neurons. Yeah.
So negative regulation really means you're in expression, or is it just actually what's
If you delete something and expression increases, then it was a negative regulator.
okay? So it's I know you don't like opposites, but
Yeah.
For me, I see, so we all look at minus 200 plus 1.
Yep.
We will still have zero expression. We mutate like.
We're not mutating anything in the minus 200 to the plus 1. We're just putting the regular minus 200 to plus 1. We're deleting everything upstream.
Oh, okay.
Yeah. Okay. So we've determined from that last one that the core promoter is in the region of 3, right? It's specifically in the region of 3 that doesn't overlap with 2 and 4. So it's in the minus 150 to the minus 175. So what core promoter is this going to be? Yeah. CPG island, just because of the distance, right? This is far enough away that it can't be a Tata box, it can't be an initiator, and an enhancer is not a core promoter, right? So that's why it's not D.
The initiator is usually plus 1.
Yes, the initiator is right at the start of transcription. Yeah. The enhancers can be almost anywhere. Enhancers can be very far upstream, very far downstream, but they're not core promoters. Deleting an enhancer will not drive your transcription to 0. Yeah. It's always in that range for this class. Minus 25 to minus 35. Cool. Yeah. We can determine that it's the range of 3 that doesn't overlap with 2 and 4, right? Because If it was in the minus 150 onward region of 3, it would also be destroyed in 4. If it was in the minus 175 and upstream region of 3, it would also be destroyed in 2. But 2 and 4 are fine. So we know it's just in this overlap region.
So distance-wise, it's like initiator closest, furthest apart.
Yes. Okay, so now we're going to do a DNA's footprinting assay. So I wanted to draw out a little bit of how this works, see how well this goes. So you can think about it, looking at your labeling, right? You have your DNA. And you have your label at one end, right? And if there's no proteins binding, you're just going to get digestion everywhere. So you can think that you'll get every possible base pair thereabouts as a band in the gel, right? You'll have this one base pair cut, 2 base pair cut, 3 base pair cut, so on and so forth for the whole DNA, right? So that's what the no protein will always look like. And then bands everywhere for no protein. And then, if you look at a protein that's binding, right, and remember, your radio label is at one end, right? If you have a protein binding here, you're still going to get these cuts and these cuts, right? So you're still going to get 1, 2, 3, 4, 5, like you're still going to get all these short cuts. You're going to be missing a longer cut if it's binding towards the end. If it's binding towards the beginning, you'll get longer cuts still, but you'll be missing shorter cuts, right? So this one, you'll miss longer fragments. So you'll have like the very longest fragment still, maybe a few more, but you'll miss some long ones in the middle. And then with this one, you'll be missing the shortest fragments. So you'll have something like this. And then a blank at the end of the gel, right? So this tells you what region of, what region of the DNA was protected by the protein that you added in the test tube, right? So we protected the DNA, at least in the case of the first one that I've drawn there, we've protected some of the longer fragments from being made, and we've protected some of the shorter fragments from being made in the second condition. Any questions on the basic concept of this before we get going into it? Yeah? Yes, it binds so that the DNAs can't cut it, right? So it's protected from DNAs. It's like the DNase protection assay. It's another way of doing it for looking at DNase footprinting. Yeah? Yeah. In reality, these gels won't even look this neat. This is honestly probably our artifact of how she had to make this in like Word or whatever. In reality, these gels have like staggered gaps that don't make a ton of sense, and that's usually just because of the way the DNA itself folds in the test tube. DNAs can't perfectly cleave between every base pair. Okay, so we have our Huntington's promoter region. We've labeled the minus 150 end, right? And then we've added protein A by itself, protein B by itself, and protein A and B together, right? So part A, protein B binds to position minus 50 to minus 25. Is that going to be true? I'm seeing one nod, and I'm going to take the nod because, yes, if the protein binds right here, you are protecting the longer fragments. You are protecting the fragments that are going to be 100 base pairs going all the way to 125 base pairs, right? You have to do your subtraction from the minus 150. You are protecting making fragments in this region. You have to flip it. You have to flip it for what's actually in the gel, right? So if the protein binds here, you're no longer cutting that with DNAs. You're going to have a gap in the footprint at the 100 base pair to 125 base pair region.
That's why C is incorrect.
Yes, we'll get to C. Yes? Yes, that's exactly correct. Yep. You look at the gel, you count 100 base pairs away from where your label is, and that's where you start. Yeah.
So do we flip the gel to the left or right? Because we're trying to flip it to the left so the marker matches up with the 150 or no?
If you really want to like flip it visually, you would flip this promoter, right? This thing, you would flip it vertically. Like if you wanted to align the promoter with the gel itself, you would draw it like this, where this is the minus 150 and this is the plus 1. Because it's size, right? So like if I'm blocking in this region, right, I'm protecting fragments that should be this size, this distance.
So the tag or like the label should be at the bottom if we're going to look at it.
If you're going to look at it that way, yeah. Yeah. You can totally do that if you want to. You will always know where the label is. It will always be written on any exam. You would have to interpret it in the reverse orientation. Okay, so B does bind in position minus 50 to minus 25. How many base pairs of the promoter DNA are protected by protein B? 25, right? So the region between where that footprint is, from 100 to 125, 25 base pairs are protected. Protein B binds to position minus 100 to minus 125. That is not correct. There's nowhere on there, or there's no footprint on there for just protein B. And then finally with D, protein B requires protein A to be bound. No, it's reversed, exactly. Protein A doesn't bind by itself, but does bind when you have protein B. Yeah? The IMSA. When you do the IMSA, the electromobility shift assay, you can tell that it is binding a piece of DNA, but not where in the DNA it's binding. Yeah. Exactly. Yep. Yeah. So, yes. Protein B protects 25 base pairs of this DNA from the DNase treatment. All right. Okay. So, based on the results of this IMSA, using radio-labeled HTT promoter as the probe and the indicated proteins, which of the following can be concluded? Protein A requires protein B to bind the Huntington's promoter DNA? Yes, right? So this is just another way of showing what we just showed previously with the DNase footprinting. When you have protein A by itself, this does not upshift. So this is a non-denaturing gel. The protein remains stuck to the DNA. That will make it heavier. That will make it more massive. It will have more difficulty getting through the gel. It will run slower, right? So if the protein is bound, it will run slower, so it'll be near the top of the gel. Without any protein, you see the no protein band, or the no protein lane. The DNA is all the way to the bottom. For B, protein B binds the promoter DNA. Yes, so we see our upshift, right? For C, protein A plus B results in the highest level of transcription. We don't know anything about transcription here, right? We just know that they bind. We don't even know if they're activators or repressors or anything that they're doing here. We just know that they can bind the promoter. And then finally, D, protein B inhibits protein A from binding. That's the opposite of A, right? So clearly, protein A requires protein B to be present in order to bind. That's why there's a further upshift only when they're both together, right? D is the opposite of that. D would be if A bound by itself, and then when you added B, suddenly you got a downshift. Yeah.
So only with EMSA and the pre-pressure considering
I don't know, to be honest. I don't know how that language is going to play out on like very specific things. Just pay close attention to the wording of the question and whether it's saying, does this data support the following hypothesis or refute it, or is it consistent with it? Pay attention to that language. I don't know how it's going to be spread amongst questions. I honestly don't remember from when I took it in advance. But I don't think there is anything in the exam that is made specifically to trick you like that, as far as I can remember. Yes. Yeah. Yes. So because protein B by itself leads to an upshift, there's binding there. Thought I saw. Yes. It's not how well it binds, it's just how big the protein is. Yeah. So a bigger protein will cause a higher upshift. Yeah. Okay. Below are the results of an in vivo reporter assay. So this is where you put a plasmid into cells that is expressing these proteins, or a reporter that, you put plasmids in the cell that express these proteins, as well as a reporter that contains the brain, the BDNF gene promoter. So that's your promoter, and you add or don't include element H as needed to do this experiment. So what can we conclude from the levels of expression that these cells give when we add different amounts of Huntington's repeats and plus minus element H? So Huntington with 50 repeats acts as a repressor through element H. Is that true? Do we see repression with adding element H in the Huntington with 50 repeats? Yes, right? So we go from an expression of 5 to expression of 0. Huntington's with 30 repeats acts as an activator through element H. Is this also true? Yes, right? So we see expression double from 5 to 10 in that circumstance only. The number of repeats in the the Huntington gene determines how it regulates reporters containing element H. Yes, right? So we see different effects depending on the number of repeats. And then finally, the Huntington's 40 does not bind element H. Do we know that from this? No, we can't conclude whether it's binding or not. In fact, it's likely that it's probably still binding. It's just about the amount of repeats that has an effect, right? Because you can think, like, if 30 is having an effect and 50 is having an effect, they're probably both binding and doing something. Why would 40 not bind, right? Even though they're having opposite effects. Yes. Not vice versa. Yeah.
So it's only if it says it possibly remains that's something different, right? Right? So like this is talking about like the upside down, and like other ones earlier with the positive regulation, naked regulation, that's for different tests, right? Altogether.
Yes, all of it is with different tests. Like, you need to be paying attention to which experiment you're looking at and whether the results support whether something is an activator or repressor. Yeah. Yeah, I agree with you on that. Yeah, the answer key for this does just have the 3, right?
Yeah, and I think
It just doesn't tell you binding directly. This
yeah,
doesn't.
The consistent is when you can like make up a hypothesis, essentially as long as it's consistent with it. Concluded is like it definitively is known from the test.
Yep.
So in order to know if it doesn't bind to element H, you would need something like a gel shift, the EMSA, or the DNA footprinting assay.
Yeah.
Concluded is like definitive by the results shown.
Will there be any like questions where it's like referring back to question 9?
There are questions that refer to previous figures.
Okay, but not like previous. Okay.
Yeah. One has element H and one doesn't, right? So plus element H means element H is present in the system. Minus element H means that it's not. Yeah. Okay. Okay. That is so tiny. Okay, you want to do a chip experiment to look at various factors and how they interact with the Harringtonin protein and the Harringtonin promoter. I'm looking at this, looking at which promoter it is. With the BDNF promoter, right? So how do we do this experiment? Go ahead. So we're going to cross-link our cells, right? And that's going to lock DNA to proteins. Then we're going to take antibodies after we've lysed the cells and pull down our DNA of interest, which is stuck to our protein of interest, right? So we take these antibodies and we use these beads and we immunoprecipitate that out. We're going to do all these wash steps and reverse the cross-linking. And then finally, we purify the DNA and we can do quantitative PCR, right? So this is a way to look at protein binding to a particular region of DNA in cells, not just doing it in a test tube like we've done with the IMSA and the footprinting. So we do that, right? And then this on the side is all the antibodies that we've used. We've used an antibody against the, I keep wanting to say Harringtonin, sorry, that's a drug I work on that's abbreviated the same way. The Huntington gene, and then SP1, acetylated H3 at K9, methylated H3 at K9, and TBP, right? So then what types of conclusions are we going to be drawing from this? Can we conclude that drug 2 is a good candidate for activating the expression of the BDNF gene? Yes, right? So we get everything we expect to see there with activation. We get the loss of methylation, the addition of acetylation, and the presence of TBP, right? So TBP is important for actually making contact with the DNA and doing that transcription. The Huntington gene can associate with the BDNF promoter in neuronal cells. Oh, sorry. Yes, so the gene itself associates with the promoter in the absence of the drug, right? So with no drug, right, we pull down on this from the neuronal cells, we still get amplification of the BDNF promoter, which indicates that the Huntington gene is binding there, or the Huntington protein is binding there. Is drug 1 a good candidate for activating the expression? No, right? So I saw a few people shaking their heads, right? It has most of the things we'd want to see. And honestly, if we didn't do the TBP pull down, I would say yes. But we did the TBP pull down, and it shows that there's no binding of TBP during the with treatment of drug 1, which means transcription is not going to be happening. TBP is totally necessary for transcription to still be happening. So that's one extra step we did past just the methylation and acetylation, where even though the chromatin seems to be opening up, even though those marks are changing, we're not getting TBP associating. Yeah. Yeah, so B is the drug, or is the Huntington's protein associating with the promoter, right? We take our antibody, we pull down on the Huntington protein, the CAG 100 repeats, right? And then we take that DNA and we use primers that amplify up the promoter, and we get signal in the no drug condition. So that indicates that that protein is associated with that DNA. Okay. And then finally, both drugs cause the chromatin structure of the BDNF gene to become more heterochromatic. No, right? It's the opposite, right? So we lost the methylation and we got acetylated. So we're going to become more euchromatic, more open. Okay. An inhibitor binds to the DNA binding domain of the thyroid hormone heterodynamic nuclear hormone receptor and blocks its ability to bind the thyroid hormone response element. So, these nuclear hormone receptors work in a very specific way, depending on whether they're heterodimeric, or homodimeric. In my opinion, homodimers are simpler, right? Homodimers exist out in the cytoplasm as individualized proteins. You get a ligand that comes in that releases that heterodimer from its inhibitor, or that homodimer, sorry, from its inhibitor complex, allows it to dimerize and go into the nucleus and positively regulate transcription, whereas heterodynamic receptors are always in the nucleus, and they're bound to generally RXR, and collectively these repress transcription until a ligand makes its way into the nucleus, a hormone in this case, and activates expression, right? So the homodimers are out in the cytoplasm, they catch a ligand and get imported in and activate. The heterodimeric ones are already in the nucleus, are always repressing, and then as soon as ligand makes it to them, they can release, basically release their repression and begin transcription of the target gene. So in both cases, addition of the hormone leads to more expression, just through different mechanisms. Okay, so this is a heterodimeric NHR. In the presence of the inhibitor, right, so the inhibitor is blocking the DNA binding capability of a heterodimeric NHR. In the presence of the inhibitor, the thyroid hormone would still bind to the NHR. Yes or no? Yes, right? So just because it's not bound to the DNA anymore, it's still going to bind the hormone just fine. That's a completely different binding region, a completely different section of the protein. It's not bound to the DNA, right? So since it's not bound to the DNA because of the inhibitor, the binding of the hormone is not going to have any major effect, right? So in the presence of the inhibitor, thyroid hormone would not regulate gene expression. I just gave that away. No, it's not going to, right? Hormone or not, you're not going to get any regulation through that. Okay? The inhibitor would cause the thyroid hormone to repress gene expression, or the thyroid hormone NHR to repress gene expression in the absence of thyroid hormone. So this is a heterodimeric one that can no longer bind to DNA. Is it going to repress gene expression? Well, it's not repressing it, right? Because it's not binding it anymore, right? So the inhibitor is not going to cause the thyroid hormone NHR to repress gene expression. It would be expressed, right? The inhibitor is gone. And the inhibitor would cause the thyroid hormone NHR to repress gene expression in the presence of thyroid hormone. No, not that either. Yeah, sorry, I'm getting a little rundown. I'm pretty sure this one is all four, unless I've mixed something up. Okay, right.
So
A and B I already talked about.
Because the NHR cannot bind the DNA, then it cannot modulate the expression.
So B is true. Right. The thyroid hormone NHR is not going to be repressing gene expression with or without thyroid hormone in C and D. It's pretty similar.